Hi there, and thank you for joining us for UnSCRIPted. I'm Angela Kashuba, Dean of the UNC Eshelman School of Pharmacy. We have an amazing community here at the school, all working toward a shared goal of solving the world's most pressing healthcare challenges. I'm excited for you to hear the unscripted stories of our faculty, staff, students, alumni, and partners. Today, I have with me Ferguson Distinguished Professor Dr. Paul Watkins. Welcome, Paul. It's good to have you here.
Thank you for having me.
So you are a hepatologist, you're a physician, and describe the journey from your clinical practice to the research center and then to the school.
I did, of course, clinical training, full fellowship in gastroenterology, and then decided to focus on liver within that discipline. And I really intended to be a full-time clinician and actually moved to Richmond, Virginia, where I have extended family, with the idea I would go into private practice there. But then they had a required 1 year in the lab as part of an NIH training grant, and I got the bug. And now I have no real clinical responsibilities in terms of seeing patients for reimbursement, but I am involved in clinical research, studies with patients and clinical trials.
Okay. So when you were doing that 1 year of research, can you talk about the work that you were doing in the laboratory and how and the journey from the laboratory to here?
Right. Well, I went into the laboratory of a fellow that was studying why in rats, when you treat them with certain steroids, they become super rats. They're resistant to toxicities, they're resistant to cancer. And he discovered that there was a particular enzyme, which turned out to be a cytochrome P450, that was being induced, and it was really an issue of these rats metabolizing and getting rid of the toxins and carcinogens faster.
Okay.
I wanted to know, was this also true in humans? So I took the antibody he had developed to the rat liver protein and went into collaboration with a surgeon to get pieces of liver and showed there was another protein, another cytochrome P450 that reacted with the antibody, purified it, ultimately got the cDNA sequence. And that turned out to be cytochrome P450-3A4, the major pathway taken by drugs in the body. So that was a very lucky early hit that put me into the mainstream of drug metabolism, drug interactions. And I had some reason to believe the metabolism wasn't just occurring in the liver, but was also occurring in the intestine. And sure enough, the antibody lit up the same protein in the intestine, and that led to studying the role of intestinal cytochrome P450-3A4. And then with collaborators like Les Bennett at UCSF, we realized the substrates for this enzyme were often substrates also for a transporter, P-glycoprotein. So I started to study that in the intestine and the interplay, the fact that the enzyme and the transporter were essentially in cahoots in keeping things out of the body. But all the time I still kept my eye on my clinical practice, which went from general gastroenterology to hepatology, and how drugs are handled the way you want them to in the liver, and then also the way good drugs can hurt the liver. And switched over into drug-induced liver injury, which has really been my major focus over the last 15 years.
Right. Yeah, so this issue of these two proteins, cytochrome P450-3A4 and P-glycoprotein, I mean, that forms the basis of the majority of drug-drug interactions. And
very lucky, early thing. I got hooked, I just became
Yeah, it's amazing. Your initial discoveries led to just a huge explosion in research in this area.
Yeah, I can't, I was part of a group. I was the first author on this PNES paper that first sort of went out this, but it was clear I was still just a fellow, doing the work and benefiting from the surrounding people.
Yeah, and you mentioned one of the issues surrounding drugs now is liver toxicity, and that's a significant problem in drug development as well. And you created DiliSim. Can you talk a bit about that and how?
Yes, so I founded and directed the Institute for Drug Safety Sciences, which was a unique collaboration between UNC and a not-for-profit research organization in RTP. But one of the projects I started was what's called quantitative systems toxicology, which is actually converting processes that drugs can start in the liver that are bad into mathematical equations. Now, I'm not a really expert modeler by any stretch of the means, but I was able to get the resources and set up a public-private partnership, a consortium, to begin building a model that would help us understand, but ultimately predict liver safety, liability, and new drug candidates. And that's been hugely successful in my whole career. That's the thing I'm most proud of starting, and I'm still the chair of the Scientific Advisory Committee for it. And the FDA is very interested in this. They actually participated and helped fund the beginning of the consortium through a CRADA. And we got a phase 1, phase 2 small business grant actually to move from some of the ways we were getting data on molecules from express transporters and cells into the liver on a chip. The microphysiologic systems, which is the hottest thing now.
Right.
The idea of trying to create a human with various cells and flow in between the things, very hot area. And so apply this mathematical modeling of DILIsym to outputs from these microphysiologic systems.
Right. Where do you feel the organ on a chip currently sits, that field? How predictive is it of the human condition?
Yeah, it's still very much an experimental tool, I would say. There's been certain areas of progress. I think the lung on a chip that is now out of WIS Institute is really very remarkable. It's a long way to go to get all the other organs together, so you sort of recreate the whole human. So metabolites made in the liver will go to the kidney, will go to some brain tissue, et cetera.
Right.
But it's doable. It's just still very much at the experimental stage.
You were just awarded, you're the principal investigator of the Triangle CERSI. So
yes,
can you talk about, which is one of 5 organizations nationally, can you talk about what a CERSI is and what the Triangle CERSI is going to set out to do?
Yeah, well, CERSI stands for Center of Excellence in Regulatory Science and Innovation. There are 4 in existence, and they've been in existence for some time. And this was the first open competition to try to, and they said they would fund a new center. All the 4 centers were renewed, and we were the 5th center. This involves a partnership with Duke and also collaboration with NC State and NC Central, and hence the Triangle CERSI, the idea this is beyond just one institution. But as you know, we're the recipient of all the funding, and I'm the corresponding principal investigator for it. Now, in terms of what they do, this really system of CERSIs was established with the FDA recognizing not only that they can't be an expert in everything, especially at the rate science is going, but that there were gaps in knowledge. So it isn't just an issue of they're getting an expert to advise them, that as they're going forward, they realize they have problems making decisions or writing guidances because the information is just not there. So the CERSIs have created a mechanism to rapidly fund collaborations between FDA scientists and academics to address specific problems, targeted specific questions with specific deliverables, including, as the information is obtained, educating the FDA examiners on what it all means. So it's a true partnership between the Food and Drug Administration and these CERSIs. One of the projects that I believe is going to be funded has already gone to the full project submission, full application, involves how to use community pharmacies, particularly in underserved areas, to reduce the harm of opiates.
Yes.
And this is something obviously of great interest to everybody. The other one is really how do you use electronic medical records to real-world data to get a better idea of the benefit and risks of anti-cancer drugs, and using very sophisticated machine learning, artificial intelligence with reams of electronic medical data to get that kind of information, because if you can, then the FDA may say, well, you don't have to test many thousands of people in a phase 3 clinical trial, driving up the cost of drugs, delaying delivery
Yes.
to the patients who really need them, if we can monitor the drug closely once it's out in the real world.
Yeah.
And so this is a very cutting edge, exciting, the real world data
Right.
aspect now. The other thing I might say, this isn't just faculty. This will have tremendous training opportunities.
Oh, okay. So
Not
unpack
just
that for
not
us,
just
yeah.
at the postdoc and PhD level, but I think at the undergraduate level as well.
Okay.
And certainly the new masters in regulatory science is very exciting. How that will be integrated in with the Cersei and across the Cerseis in general. Education,
Yes.
what they call exchange of information, is a high priority.
Yes.
And I think we're just beginning to figure out how this will really benefit education at all levels of training.
Right. You mentioned undergrads, and we are starting our own minor in pharmaceutical sciences that's coming online to be
That's
able to
right.
reach our undergrads, and so I hope that that will also be an aspect. And that's wonderful. I want to switch gears a little bit and talk about some of the fun things that you do. So you're a sports car enthusiast, I understand.
I am. It's kind of, it's aging out of it, I'm afraid. But I own a 1992, the very first year, Dodge Viper.
Ah, you do?
Yep.
All right.
And I just...
And so why a Dodge Viper? What
well,
attracted you to
because
that
you
car?
get sort of a Ferrari with a poor man's budget, basically. It's,
Okay.
it's an old Dodge. But, it's got 400 horsepower, 6-speed manual transmission. And there's a very strong North and South Carolina Viper Association, which I belong to.
Okay.
I actually did a 5-day race car driving course out in Arizona, the
Is
Bond
that
Run,
right?
because I thought with the Virginia International Raceway close that I could sort of get into racing, because they have all
Okay.
levels and all these things.
Yeah.
But I found out there's a lot more to it than I thought. And
It's very physically demanding.
very physically demanding. This whole heel-toe shifting thing in a manual car is very difficult. And I did just track it, my car, 2 months ago, and I realized I probably shouldn't be on the track, with people who really know what they're doing. I got some scary moments, so
Oh,
I
my
think that may
goodness.
be over. But still, the shows and stuff, it's
Yeah.
good hobby.
Yeah, coming together over a love of the car. Yeah, that's great. You also scuba dive.
Yes, I'm a advanced open water scuba diver. So is my wife.
Have a favorite scuba memory?
Yeah, we scuba dived when we were much younger at Club Med, and we went down to about 100 feet, and the guide went into a cave, and as he went into the cave, he said, "You stay here." As he went into the cave, 2, I would say about 8 to 9 foot shark came out on either side. Now, it turned out they were nurse sharks, which are
Okay.
totally harmless, but we didn't know.
Right.
So my wife got away hiding behind a rock. Thank God she didn't try to go to the surface, but that
goodness.
was the most scary
Wow.
moment.
Yeah, that's intense. Well, thank you, Paul, so much for speaking with us today. It was a pleasure to talk
I
to you
enjoyed it.
about
We
the
always
research.
we go to hockey games together,
That's it.
not
That's
knowing
it.
we were sitting right in front of each other most recently.
That's right.
But
That's right.
yeah, it was great. Well, thank you.
Yeah, and congratulations again on the Triangle CERSI. It's a
It was
really
a group effort, but
wonderful
I'll take the
accomplishment.
credit.
Yeah, no, we're really excited to see how this progresses over the next 5 years.
Very excited myself.
Yeah, good.
Should be good.
And thank you all for watching. We love sharing stories of those who make the UNC Eshelman School of Pharmacy excellent. We hope you join us for the next episode of UnSCRIPted, sharing stories of people who are advancing medicine for life.